So I'd like to welcome Caitlin Kelleher and James Gibaldi. They are both from the Michael J. Fox Foundation. And first I'll introduce James. In his role as Senior Associate Director of Clinical Operations at the Fox Foundation, James manages the research operations of the Foundation's landmark study, the Parkinson's Progression Markers Initiative. James holds a master's degree in neuroscience from Columbia University and has past experience in public health and traumatic brain injury research. And Caitlin is an Associate Director of Patient Engagement at the Michael J. Fox Foundation, supporting studies funded and sponsored by the Foundation. She works to elevate the patient and care partner voice in study design, development, and execution. Caitlin has a degree in neuroscience from Columbia University and has previously worked in device development and marketing. So thank you both for coming. We like to end pretty promptly at 7:30, so I wanted to start on time. If we go a little over, it's not, that's fine, but we try to end promptly if we can. So how would you like to do this? Who's speaking first?
So yeah, thanks, Kathleen, and really thanks to everyone. It's really exciting, and we're really grateful to get in front of this group and talk about Parkinson's disease, genetics, as well as research opportunities that exist, particularly for this community. I think Caitlin and I are probably going to tag team a slide presentation just to give some background and overview on the foundation, our research vision, our sort of a high-level overview of what Parkinson's disease is, just to kind of level set everyone. And then also, again, the sort of the genetic markers that we're investigating and very interested in the research space, and how you all potentially can be involved in a really exciting research opportunity that we'll talk about in a moment. But Caitlin, anything else you wanted to add before we dive in?
I don't think so. I was just going to ask, do you want to present or me?
It would be great if you could hold the slides up.
Absolutely. So everybody will bear with me for just 1 second. Great. Can everybody see that? Yeah, wonderful.
Yes.
Okay, perfect.
Caitlin, do you want to go into, yeah, there you go. Okay.
Yep, all set. Yeah, so like James said, thank you so much for having us. We're really excited to be talking with you today and to be kicking off your JETI series for this year. In particular, I'm really interested in connecting with you all and your community. We have been talking a lot at the Foundation, especially in my team and the department, about how we can better reach people, especially folks who might be interested in learning more about the genetics of Parkinson's and the Ashkenazi Jewish community. So this is a quick agenda over what we're going to cover today. We're going to talk about the Michael J. Fox Foundation briefly, go into some basics of what Parkinson's disease is, a genetics 101 on PD, and then how you all can get involved and get your community involved. So as you may know, the Michael J. Fox Foundation is the world's largest nonprofit funder of PD research. And our goal is to find a cure and better treatments for Parkinson's by aggressively funding research to develop better improved therapies for people living with Parkinson's. And the real end goal is to make sure that any and all of those treatments get into patient hands. We want to not only be funding better treatments, but bridging the gap between research, clinical development, and patients and their families. These are some quick stats about us. We were founded about 22 years ago. We're unusual in the fact that we are a public charity, but we operate without any endowment. So all of the money that we raise in 1 year, we do spend down. We don't have anything left over. We're also a little bit unusual in the fact that we don't have any chapters. Our team is almost entirely based in New York City, and we are right now about 200 people. That said, we do have a large number of grassroots fundraisers. So last year, there were over 8,000 people in that community, which is pretty enormous. And to date, we at the Foundation based on those grassroots fundraisers and then other donations from people like you, as well as industry donations, have been able to fund over $1 billion in research programs dedicated entirely to Parkinson's disease and Parkinsonisms. Right now, we are working on over 800 different projects. That is a quarter of the projects that we funded to date, and almost a third of all of the projects that we fund are led by researchers outside of the United States. And that's really important to us because when we find a cure for Parkinson's and when we find these better treatments, we want to make sure that it's not only applicable to people who are in the United States or of a particular community. We want to make sure that it's applicable across the board. Included in that mission is that all of our funding is agnostic. So we don't only support academics, we don't only support biotech and pharma, we support all of those different people who are working towards finding better treatments. So what is our role? We are really working on driving drug development. So you can see here on this chart that the US government funds a lot of discovery research, and then we do everything in between there up to late stage projects. So essentially what that means is that we're supporting these early stage and middle stage folks who are working in this development pipeline. So working to make better drugs, to make better treatment options, and helping them build up a database and data and publications so that then they can be funded by people with really enormous resources to push them through regulatory approval with the FDA and into the market. And actually right now we are working on getting more involved in that process because, as I said, our end goal is to get all of these treatments into patient hands. And we know that regulatory approval is a really important part of that. And just because something is approved doesn't mean that it's necessarily accessible to the people who need it. And we want to make sure that we're moving things from the very beginning, from A to Z, getting to those patients. So what's going to help us move through that process and get better treatments and to get better drugs faster? And the main thing here is that we're looking for biomarkers. And a biomarker at the most basic level is a biological characteristic, that can tell us more about somebody's risk for developing a disease, and then when that disease might develop, and then how it might change over time. So a good example of this is cholesterol level for heart disease. People with higher cholesterol are often at higher risk for heart disease. If it reaches a certain level, they're likely to develop heart disease. And then you can use their cholesterol level as a marker for how their heart disease might change over time. So the issue with Parkinson's, or one of the many issues with treating for Parkinson's, is that there are no validated biomarkers. And we're really working to find these sorts of tools because that will help us to diagnose Parkinson's earlier, chart the disease progression, design clinical trials to make even better treatments, and then test the impact of new therapies. And Parkinson's is an unusual degenerative disease in that it is both heterogeneous and nonlinear. So that means that it presents very differently in every person who has it, or it can present very differently in every person who has it. And somebody's neurodegeneration or decline can be very different from year to year. They could have a number of really good years and then a few bad years, and vice versa. So researchers are trying to find these sorts of biomarkers so that we can better chart disease progression and understand whether somebody will develop Parkinson's at all. Examples of things that we might investigate for that are...
Are you close to finding any of those biomarkers?
Great question. James is going to talk a little bit more about that when we get into PPMI. I think the answer is we do have some genes that we know increase your risk for PD, but unfortunately, it doesn't always mean, or not unfortunately, it's very good that it doesn't always mean that you're going to develop PD, but there's no one answer. James,
yeah,
do you have more?
There's a lot of really good early signal, I would say, for many of these biomarkers. So when we talk about what PPMI, Parkinson's Progression Markers Initiative, what data is coming out of that, I can speak more to sort of what the early signs are showing us. There's a lot of promising data coming out, but no, the answer is we don't have a single biomarker yet for Parkinson's, which is, I think, exactly what we're trying to look for in this research.
Yeah, great question. Thank you, Mr. Levine. So PPMI, which James just referenced, is the Parkinson's Progression Markers Initiative. This is our landmark clinical study to try to understand how Parkinson's disease starts and then changes over time. And the goal here is to identify indicators or biomarkers of PD progression so that we can get to that earlier diagnosis, improve disease tracking that will help folks better manage their treatment regimens once they're being treated, and then more efficient testing of those new therapies to improve the odds of success, and then also make it so that getting treatments to market costs less and is much faster. And PPMI is an observational study, which means that participants are not given any sort of drug or any other type of treatment as an intervention. We're purely collecting data and looking at it over time and comparing it between a number of different groups of people. Right now, it is happening at over 50 sites worldwide, and that's always expanding. We have a goal of enrolling 4,000 people by the end of next year, so by 2024. And then we also have a goal to enroll 100,000 or more people in an online portion of the study. And what we're here to talk about today, and what we're going to talk about a little bit more now, is we're really trying to find people who might carry certain genetic mutations that are associated with an increased risk of PD to better understand those markers in Parkinson's disease. So now that we've talked about the foundation, James is going to give you a bit of a background on what Parkinson's disease is, and then some genetics of Parkinson's disease.
So before we move on to this, are there any questions on anything that Caitlin presented?
Yes, I have a question in terms of, she said that in the observational studies, no meds are given. Did she mean no meds of any kind or no experimental meds, but they're continued on treatment as usual?
Great question.
That is a great question.
In this observational study, we primarily are looking at people who are not taking meds at all, who have not yet had to start taking medication. However, because they're in the study for so long, once they start taking medication for any symptoms that they're having, they continue to take those. It means that we're not giving them any experimental meds.
Okay. And the other question, did I hear correctly? Did you say that my impression of Parkinson's, I haven't had that many patients with it, but my impression of Parkinson's is it is at varying speeds continuously progressive. Did you say that there might be several years of good functioning, then bad, then good?
Yeah, so that's,
yeah, James, go ahead.
It's such a heterogeneous disease in that the experience is really unique. And I think one of the things that we are looking for in PPMI or studying in terms of progression markers is what predicts someone to progress differently than someone else. And so I think, what makes someone progress with good function for, a number of years and then sort of more rapidly decline or decline slowly over the course of 20 plus years. Those are all things that we are trying to study by collecting all of these data. Great questions, though. Any other questions before we kind of get into the basics of what Parkinson's disease looks like clinically? Okay.
I have a question.
Sure.
How long, what is your hope for the longevity of these follow-ups? I mean, when somebody agrees to this, how long are they agreeing to be part of your study?
Great question. It is variable. We generally hope that people are in the study for at least 5 years, but folks who joined the study in 2010 are still in it. So coming up on 13 years now.
Well, the longer the better, right?
Absolutely. Yes. If we could study people for like 25 years, that would be amazing.
So to just get into some of the basics here, in terms of how many people we're talking about with PD, there's about 10 million people worldwide that we know of that have Parkinson's disease. And I say that we know of because I think, Parkinson's is the type of disease where people could have symptoms for many years without sort of knowing that they have PD and come into the clinic. And so even understanding that number can be a little bit trickier than you'd think. Parkinson's is the second most common brain disease after Alzheimer's, and 1 in 100 people over age 60 will develop Parkinson's. Though there are rare cases, someone like Michael J. Fox, even who are diagnosed at age 40 or younger. So there is a subset of the population that develops young onset PD, although we do see that it usually appears after the age of 60. And Parkinson's is a neurodegenerative disease. What that means is that over time you start to lose, you lose neurons and neurological function, and motor symptoms, which are really the classic hallmark signs of Parkinson's. This is your resting tremor, your slowness of movement or rigidity, issues with balance. And then there's a lot of non-motor symptoms, I think, that are less talked about. Cognitive dysfunction, depression, anxiety, constipation and digestion issues, and sleep disorders. And actually what we find is that these non-motor symptoms can go undetected as Parkinson's symptoms for really decades or years. And actually what we're trying to do with PPMI now is understand these non-motor symptoms so that we can understand who could potentially develop the motor symptoms over time, so that we can better predict progression and risk. So what causes Parkinson's? I think this is sort of the crux of the conversation today when we're talking about genetics. But really the majority of those diagnosed with Parkinson's really fall on a continuum. And it's really a mix of environmental factors and genetic factors, with some populations due more to genetics, and really some populations really is coming from exposure to environmental factors like pesticides that are highly studied, paraquat, for example, or head trauma, which is still sort of in the earlier stages of investigation, but is something we know is linked to Parkinson's and correlated. Usually, as you can see here, it's sort of a blending of the two, and we can talk about sort of what this looks like in the next slide with some of the genes. Before we get to that, actually, the treatment landscape. So what are we doing right now, or what's sort of the standard course of treatment right now for those that actually develop Parkinson's? And many of these treatments have really gone unchanged for a while. And so there's a lot of new therapies coming to the market to help specific symptoms, but largely the course of treatment remains the same. You're trying to replace all that lost dopamine, because again, it is a neurodegenerative disorder, so you're losing dopamine over time. So levodopa is something you'll hear a lot as a treatment. That's replacing the lost dopamine. Carbidopa is actually just a compound that helps the levodopa get absorbed more into the brain. And so oftentimes you'll see the two sort of stacked together as a treatment. And then you also have, it's also something important to note that levodopa really loses efficacy over time. The body, over time will develop, will sort of become tolerant to it over time, and you'll start to wane in its effectiveness, which will then lead to sort of a deadened effect, as well as side effects that it imposes, such as dyskinesia, which is sort of the classic sort of wild movement that you see with Parkinson's. There are surgical procedures. Yeah, go ahead, Bruce.
Yes. Can I ask you, what is dopamine?
So dopamine is a neurotransmitter in the brain that promotes movement, and also can can also regulate mood as well. And so it's a very important neurotransmitter. And when the balance of dopamine, or sort of the loss of dopamine in key areas of the brain takes place, it really affects movement as well as mood. Caitlin, anything else you wanted to add to what dopamine can do?
I don't think so. I think that covers it. Basically here, the loss of dopamine leads to all of those different effects that James was talking about. And I think oftentimes we associate Parkinson's with only motor function, but because it is primarily dopamine loss, it also affects your emotional function and other aspects.
Thank you.
Of course. And so there are some surgical procedures that are, that have become, I think, increasingly utilized. One of them being deep brain stimulation, which is when they're actually able to surgically implant a stimulator into your brain to sort of pump the dopamine or the levodopa into your brain on a more regular and sort of regulated way. Now, not everyone's eligible for that treatment. And I will say that again, sort of over time, it becomes less and less effective, although it is a more effective way of sort of bringing that dopamine into the brain rather than sort of taking oral pills. But again, it also only treats the motor symptoms.
I actually have a question.
Sure.
I know some Parkinson's patients have engaged in vigorous physical activity, which has helped them to forestall taking the Ldopa. Have you looked at that?
Yes. So there are many like exercise treatments that we definitely see our patients utilize, and a lot of people anecdotally say that it's great for them psychologically as well as physically. It is a great way of sort of delaying needing levodopa, because once you start down that path, then again, like I said, it sort of wanes over time. Caitlin, did you want to speak at all about sort of like Rocksteady or anything like that?
Yeah, I think that's a really great point, Kathleen. And we specifically do have some partnerships actually with the gyms that have classes specifically for people with Parkinson's disease, because some of those exercises are modified, but it is often a much more rigorous exercise than you might expect. So James just mentioned Rocksteady boxing. There are boxing classes that are adapted for people with Parkinson's all across the spectrum of disease severity. And it does oftentimes help put a bit of a pause on you having to take levodopa. I think another thing we also get questions about all the time is diet, and diet can help with medication management and mood. We all know gut health, brain health is connected at this point, but in the long run, most people are going to have to take either a medication like levodopa, carbidopa, or have a surgical procedure within a fairly short period of time, just depending on their disease progression. Sandy and Joel, I see that you're raising your hand.
Yeah, so I just wanted to mention another type of exercise program that's been very beneficial is a dance program for people with Parkinson's, because of the balance issues too, especially. And my other point is, and correct me if I'm wrong, with deep brain stimulation, which is a fascinating and wonderful treatment for people who need it. The side effect sometimes is decreased cognitive function after deep brain stimulation. I'm not sure if that's what you found or whether you agree with that.
I'm not as familiar with the side effects of DBS, but that certainly could be the case.
Yeah, I think that with any invasive procedure, you definitely run the risk of that. I think that it's probably, again, case by case. Personally, we know a lot of folks who've had DBS, and I don't know if they've seen a marked cognitive function decline, but it certainly is possible. It also could depend on when they have the surgery, at what point in their disease progression and their age. Great.
Okay, so now really diving into the genetics. So again, the cause of PD is really unknown, right? And so that's really what we're looking for when we're searching for these biomarkers as well, is sort of not only what are the biomarkers, but what can cause these biomarkers. Genetics and environmental factors play a role, as I mentioned in the 2 slides ago. However, we do know that about 5 to 10% of Parkinson's cases are linked to genetic causes, which is quite low. A lot of times, people really think of PD as a genetic disease, and the reality is it's really a mix of environments and genetics. However, certain genetic mutations do account for more cases of PD in some populations. And so I think especially for the Ashkenazi Jewish population, LRRK2 and GBA, these are two genetic mutations. These are more prevalent in the Ashkenazi Jewish population. And so that does increase, if you do have those genetic variants, it does increase your risk of developing Parkinson's, but not markedly. So estimates vary, but about 15 to 20% of Parkinson's cases in this population are attributed to LRRK2, and 20% are attributed to GBA. And you may have also heard of Gaucher disease. That's also linked to GBA as well. And so really, these two genes are highly studied, especially in PPMI. And I think Caitlin's also going to tell why studying these genes in the Ashkenazi Jewish population is so critical to understanding the root causes of Parkinson's, really because this is sort of an enriched population for these genetic markers.
Great. Yeah, so specifically, the question that I'm sure you all have and that we often ask ourselves is, why are these particular genes so important to PD research? And it's because we've found that, and researchers have found that mutations in both of these genes, so they're two different genes, but separately, they're related to both people getting and then the progression of Parkinson's disease. And I really want to emphasize here that just because you have this genetic mutation does not mean that you're going to develop Parkinson's disease. And the majority of people who have either of these do not go on to develop PD. And so then by studying people who both have and don't have Parkinson's disease.
Start video.
Who have those genetic mutations means that people can understand better why some folks go on to develop Parkinson's disease, some folks don't. Yes, Sue, do you have a question I can answer now, or that might have been a mistake? Okay. We can also use those genetic mutation differences to better understand the more subtle or nuanced symptoms of Parkinson's disease. So some of them, those non-motor symptoms, for example, or a very minor resting tremor, things that we might not recognize as being a type or a symptom of PD. And then we can also better develop and understand new treatments for everybody with Parkinson's disease, not only those with genetic forms, if we're studying folks who both have it and don't have the genetic forms. And then the sort of larger picture here is that we're trying to further this sort of genetics revolution that is happening in Parkinson's disease research right now in pursuit of a cure. Yes, Sue, is your hand raised, or is that?
Yes. Go ahead.
Okay.
Okay.
Okay. What I was going to say was, you mentioned the Ashkenazic, but I'm pretty sure I'm Sephardic, even though it was not confirmed, because my father's side of the family went to Holland and Belgium, I think after they were in Spain. And what I'm trying to say to you is that, like, I had an aunt and an uncle who had Parkinson's. My uncle got it when he was 40. Mm-hmm. And then my grandfather had it also, and my father. So hoping I don't get it, but that's because you mentioned Ashkenazic, so it sounds like it must be in the Sephardic population also. Is there any data on that?
So I think,
yeah, go ahead, James.
Well, I was just going to say, I think that I have gotten that question before. Obviously, with the Ashkenazi or the Sephardic population, I think you could see these genetic mutations. You could see these genetic mutations in other populations as well. I think we just wanted to highlight that in the Ashkenazi population specifically, the numbers are just much higher. But I would certainly say that, until you get a genetic testing, you really don't know for sure if you do or don't have LRRK2 or GBA.
All right. Thank you.
Great
Thanks for the question. Oh, Barbara.
Yeah, thank you. So I was wondering, I understand the concept of risk factor. Is there been any work done about increased or unchanged risk, whether you have inherited either one of these mutations from both your parents or a single parent? Is there anything known about that? Does it change the risk? Or is that too hard to find?
I think
That's a good
down the
question.
line. Yeah.
Go ahead, Kate.
I was going to say, I think that's a great question. I think that's one of the things that we're trying to get at with this probably as well, is the more data that we have on people who have these mutations, whether they have essentially like 2 genes, so like the entire gene pair has a mutation, or if it's just on 1 gene, then that would mean that you received it from either like both parents or 1, right? But as of right now, I don't think that we have research that's that granular on it. We're still compiling this data set. James, do you have anything else on that?
And to follow up, is this mutation in a single place in the protein, or is it basically, are there many mutations that confer increased risk?
So I guess, are you asking about specific variants within the mutation, or?
Yeah, sure. I mean, are many amino acids subject to being changed, or is it a single locus on the protein that changes? You know?
I believe it's I can't say with confidence either way. But I do know that there are specific forms that LRRK2 can take, or GBA, they are looking at. So more specifically, LRRK2 G2019S versus G1441.
Thank you.
Great question. I love it when we get questions that we can't fully answer. It means that we have more that we need to learn and do and look at. Great, so that covers, I know that we just tried to cover a lot of very complex genetic and gene information in this slide. And now that we've talked about that and why this particular population and anybody really is important to be involved in this research, we're going to talk a little bit about how you can get involved. So currently, we're looking for individuals who meet certain criteria who would then go on to receive these special genetic test, or not special genetic testing, to receive genetic testing. And then
they're talking about the
Could you please mute whoever just joined us, please?
Great. Yeah, so essentially we're just looking for a number of different people to join the study. And this is more specifically these populations of interest because of those genetic mutations we were talking about that put you at higher risk for developing Parkinson's disease. This is also to say that we would love for all of you to join the study if you would like to, and I'll talk about that a little bit later. But the 2 categories that we're really interested in are people who either have Parkinson's disease, so who have been diagnosed with Parkinson's disease, who are of Ashkenazi Jewish ancestry, are 30 years or older, and have a diagnosis of less than 2 years. So meaning if they're 32, they were diagnosed at 30 or older. We're also looking for people who don't have Parkinson's disease, who are also of Ashkenazi Jewish ancestry. And in this population, we're looking for a slightly older group. So folks who are 60 years or older and have a first-degree relative. So that means a parent, a child, a sibling who has Parkinson's disease. So I think some of you on this call might be very good candidates for this. And we're studying folks with these types of mutations who either have Parkinson's or don't have Parkinson's, because it's going to help teach us about the biology and help us try to look for those biomarkers that we've been talking about, and then accelerate research towards more effective therapies. So this is the website there, and I'm going to say this a lot of times now for the rest of this presentation. It's www.michaeljfox.org/PPMI. And you can go there to learn more about the specific study and how things will go. This is what participation would involve. Essentially, you just go to either the website that's down there or ppmionline.org. You take a quick survey to find out if you might qualify for genetic testing for no cost. You'll complete a DNA test via mail. So it's just like a saliva sample. And then if you do this genetic testing, then you'll receive genetic counseling with no cost to review the results of that test. And then based on the results, you might be asked to participate in more tests and assessments for this biomarker research over the course of a number of years. You can also participate even if you don't have these genetic markers or even if you don't have this ancestry by just going to the ppmionline.org and you can participate online. And the reason that we're asking this of you to consider and of essentially anyone in your community is that only patients and families can teach us about a disease. We can ask all of the questions that we want, but if people don't raise their hand to participate, then we don't have the data that we need and we cannot move forward with developing better treatments. And hundreds of people are needed for studies. In this case, we're looking for thousands of people. And the standard rate of recruitment for trials is only 1 person per site per month. So that would be 12 people over the course of an entire year. And the question that we're always asking is, what would happen if we were able to speed that process up? What would happen if we were able to get it to be 2 people per month or 3 people per month or even 4 people per month? And the more volunteers that we do have enrolled in studies, the faster we're going to have new treatments, new insights, better understanding to answer all of your wonderful questions, and eventually a cure. This is just a breakdown of how you can get involved with the different links, the phone number that you can call. And then I do also want to just say, if PPMI is not of interest to you personally, or if it's something that you're not eligible for, there are lots of other ways to get involved with Parkinson's or atypical PD trials, and you can do that at this other website. And there are hundreds of studies listed there that anybody might be eligible for in your area, in Connecticut, or around the world. So thank you again so much for having us. We have really enjoyed sharing all this with you and would love to answer any other questions that you might have.
Thank you so much. And if you could stop sharing your screen, we can see people, and I'll change the view. So does anybody have a question for Caitlin or James?
Yeah, I have a question. My father had Parkinson's, succumbed to it after dealing with it for about 25 years. My sister and I were in a genetic study, which I think was through the University of Indiana a couple of years ago. More than a couple of years ago. I don't wonder if this is an extension of the same study you're doing. It was a genetic study on Parkinson's?
Yes, that sounds like it would probably be similar because the genetic testing that we do in the counseling with PPMI is through Indiana University. Okay. And so if you've worked with them, it's yes. Same group. Yep.
Okay. Thank you.
Roberta, yes, I see your hand is raised.
How old can somebody be to be a volunteer? I have a sister, a younger sister who has Parkinson's, and I'm in my late 80s. Would that be acceptable for the study?
Yes, you can absolutely volunteer. I don't think that we have an age limit on the upper end. It's just the age limit on the lower end.
Okay, thank you.
Yeah, absolutely.
Well, you have at least one volunteer from Atlanta.
Oh, great. Delightful.
Give you a call.
Please do.
So I have another question. You said that you can't be on any medication. Does that include Asplect, which is like the first medication that they give to Parkinson's patients? Or is it just the Ldopa medication that you can't be on?
It's it's basically most common PD medications. I don't know them off the top of my head, but it would likely mean that you were not eligible to participate in clinic, but you could still absolutely participate in the online portion, which is really important to us as well.
Thank you. That's a good distinction. Thank you.
Yeah, of course.
So we have Bruce and Nancy.
Go ahead, Nancy.
Okay, just 2 quickies. Did I hear you say in the beginning that this is the most common after dementia for older people across the board?
After Alzheimer's.
Right, okay.
Yeah.
Also, you mentioned, different people respond differently, in terms of the progression. And I was thinking of a friend who had MS, and that was also the same situation. So are they connected, or do all diseases behave this way? Or many diseases behave this way?
That's a great question. I think my answer to this would be I'm not a neuroscientist, but a lot of neurodegenerative disorders do have a nonlinear progression because they affect the brain in different ways for everybody. And so the degeneration could be really fast in some people or slower in others. So although they affect different systems, MS and Parkinson's disease, they are similar in that way.
Okay, thank you.
Bruce.
Yeah, I'm just curious, and if it's proper for you to answer, and if you can answer, how is Michael J. Fox doing?
He's hanging in there. He's had Parkinson's for over 22 years at this point. So I think his spirits are great, and he still shows up to board meetings as fired up as ever, and is really, just impatient to get a cure and treatments through. And so he's very inspiring as the leader of our foundation. And I think he's doing as well as he can.
Sandy has a question.
First of all, I think this is a wonderful study, and it's very noble for people to volunteer and be involved in this. But I'm sure there's a difficult question when it comes to doing genetics early on in your life, as far as since we don't have a preventative treatment for Parkinson's, and some of the earlier therapies really lose their efficacy later. What would be the benefit of doing genetics sometimes? Wouldn't it cause more anxiety in people to know what the future would bring? Also, there are certain tests now for Alzheimer's as well to try to pick it up early. People who are younger, who need health insurance or long-term care insurance, might not have that available to them if that diagnosis is there. So I'd like your comments on that if possible. Thanks.
Sure. So I think there's a few things to touch on there. So first of all, I think one of the items that you touched on really highlights the importance of genetic counseling, which is a part of this study. So when you have the genetic testing, you also have genetic counseling to really understand, what does this mean for you? What does this mean for your future? What does this mean for the rest of your family? And so to be able to talk to real genetic counselors rather than just sort of be given results with no context, I think is very important, and something that we're always advocating for, both within this study and more generally in research. Context around results is really key. I think, and the first point you made about prevention, I think, is actually a real goal of PPMI. PPMI expanded in 2020, and our goal now is really to move earlier into that risk space, to understand risk more, because pharmaceutical companies that we're talking to are actually interested in potentially developing preventative therapies. So I think that's sort of the next horizon here, is preventative therapies. But before we get there, we need to understand risk and who has certain risk scores and then develops Parkinson's. That will then inform the prevention treatments. And like you said, in Alzheimer's, they're also doing this. And I think they're a few years ahead, as they always are, a few years ahead of where Parkinson's is in sort of that prevention space. Barbara?
Yeah, so my question's a little bit along the scientific lines, just because I always thought that Ldopa, and I could be wrong about this because you implied something else, that basically it stopped working because as the dopaminergic neurons died, then they lost their target. that the Ldopa had no place to act because the neurons were dead, but you implied it was, something a little different than that. And I was wondering if you had any information you could offer on that.
It's certainly both. A large portion of that is the fact that there are less neurons for the Ldopa to actually bind to, and you're sort of racing against that neurodegeneration by just sort of pumping in the Ldopa. But I think like any other drug, the efficacy does wane over time as your body sort of becomes more tolerant to it. So it's definitely both. So thanks for highlighting that.
Yeah, so thank you.
These are great questions. Any other questions?
Well, if not, we'd really like to thank you. This has been.
Debbie Schwell has a question.
Sorry, don't want to cut anybody off. Go ahead, Deb.
Muted, Deb.
Okay, now that I'm unmuted, this is a little bit out of the focus of your talk, but I have read that rotenone, which is a common organic pesticide, can impact upon Parkinson's. And the reason I'm asking is that my father died of Parkinson's and he had used rotenone liberally for about 50 years. And there's a dual factor, possibly the Ashkenazic connection, but also that his extreme exposure to this one pesticide. And it wasn't clear to me that your organization does any kind of research outside of the genetic field, but I was just wondering if you had any comments about that.
Yeah, so certainly we are investigating environmental factors, including pesticides. Pesticides are, I think, #1 on the list of environmental factors that we are studying. We also do ask, as part of participation in the study, we ask all participants questionnaires about exposure to certain pesticides, or potentially, if they're vets and they worked with Agent Orange in Vietnam, something like that. Or again, if they've used paraquat and worked in the agriculture industry. These are all questions that we try to ascertain from our participants, and also we fund policy in Washington as well as a foundation to try to ban the use of a lot of these pesticides.
Dan has a question.
Thank you, Bruce. A question in terms of if people, let's say, know they have the genes, getting back to Sandy's point, are there lifestyle modifications? For example, given the growing evidence of alcohol's diffuse toxicity, as the World Health Organization is pointing out, if somebody knows they have, does alcohol have any bearing on the progression of the illness one way or the other?
I'm not familiar with research in Parkinson's and alcohol use, and I think that there are there's a lot of sort of anecdotal evidence for what people do to try to sort of mitigate their risk. We work a lot with folks that have something called REM sleep behavior disorder, which is a sleep disorder where you act out your dreams rather violently at night, and that puts you at high risk for developing Parkinson's. And so when we talk to folks that have this condition, they do talk about sort of, steps that they take to try to prevent what they see as, Parkinson's coming in the future. But there's not a lot of great data right now that shows that is successful. I think that will sort of come as we understand risk a lot better.
Well, thank you all for.
Just a quick point. It's interesting because alcohol is such a diffusely used potential neurotoxin. Mm-hmm. It's interesting that it hasn't been looked at that much.
Yes, I think I think we might ask about it as part of our more like upfront questionnaires, the same way that we ask about potential occupational exposure to pesticides. We might ask about sort of like daily or weekly alcohol intake. But beyond that, I don't think we do. Yeah.
So I just want to make a comment on Dan. You know, in benign essential tremors, sometimes alcohol or beta blockers actually are beneficial in small quantity, of course. The problem, I think, as well as treatments in Parkinson's, is you're dealing with things going through the blood-brain barrier. And sometimes some of the treatments are effective in certain people with Parkinson's and not in others. And they can't figure out why, but they think it's because in some people, the blood-brain barrier acts differently. Maybe you know some of this too, but that's some of the research ongoing. One of the members of my family is doing that research actually. And therefore, they find that certain drugs will go through and get to the areas involved and others won't. So that's alcohol would probably be the similar case. Yeah.
Well, thank you for the very important work.
Oh,
one more.
Nancy, go ahead.
Yeah, I would just like to ask Caitlin and James how they got involved in this. Like, you know, how did it happen?
Oh, great question. It's like a job interview. How did I get involved here? I joined the foundation fairly recently. I joined about 6 months ago. And before this, I was doing a pretty big mix of things. I was a clinical research coordinator in psychiatry at Mount Sinai, a hospital in New York. And then I moved into industry for a while. So I was working on device development for alcohol use disorder and opioid addiction. And then I worked at a mental health startup for a little while, connecting therapists with people. And then I came to the foundation because I wanted to get back to research without honestly having to perform the research. It's really nice to be able to work with so many people who are so interested in these research questions and be able to support them and move towards doing something that I think is really important in the long run. James?
Yeah, so I can share quickly. I've been at the foundation nearly 3 years. And before this, I was actually at City Hall. I was in the mayor's office for a couple of years working in public health. Before that, I was at Weill Cornell's Department of Neurosurgery as a research assistant in neuropsychology, really focused on concussion research and clinical assessments done for actually a lot of athletes like the Jets and the Islanders. So that was a good time. So yeah, I think how I came to the foundation was really just I have a big interest in sort of how sort of neurodegeneration and specifically my research was on neuroinflammation, but how neurodegeneration in these various illnesses manifests itself. And so I think there was a real draw to the mission of the foundation and specifically getting involved in the clinical research end of things.
Okay, so I don't see any more questions, but thank you again for this incredibly important work that you do that, you know, if you're successful, will impact many of the people and the families associated with the people on this call. So we're very grateful. It was a very interesting presentation, and we look forward to hearing good news from you in the future.
Thank you so much for having us. We really appreciate it.
Yeah, really appreciate everyone's time. Hope everyone has a good night.
Take care now.
Thank you.
Bye-bye.